if we break the hydrogen bonds, we can then do what? We can then see that in both directions, we can have replication. So the V structure that will form, as we see where it is opening, which will be both on the left and the right side, those sides where the replication is going towards. And so the DNA will be opening towards those ends, either on the left side or the right side. That is a fork. So F-O-R-K. So we call it a replication fork. And that is the side that the the DNA polymerase is going to follow when identifying a template strand. And this gives us what we identified yesterday as leading or lagging strand. So that replication fork is important. And the whole initiation, the recruitment of the helicase itself. So helicase loading and identification of this bidirectional, meaning two direction, to the left as well as to the right. So we have the DNA A, which recruits the DNA C. DNA C is another protein, which is a helicase loader. So it's responsible for loading what? DNA and the helicase to that the helicase itself. So let's get familiar with these terms. So you hear DNSC. What is DNSC? DNSC is a helicase loader. A loader, it means it's responsible for loading helicase. DNAB is actually the helicase enzyme itself, which is able to melt DNA or unwind DNA by breaking the hydrogen bonds. So let me repeat these. DNA, what is it? You should know what it is, what is raw. DNSC, what is it? It's a helicase loader. So what exactly does it do? Then DNAB is a helicase itself. At least know what each one of these mean. So sometimes we can use helicase enzyme or DNAB. So you should know what these mean. So now we have two DNAB hexamers. Hexamer, it means there are 6, right? We have two DNAB hexamers that are loaded, one at each side of the melted what? Origin. So because they are loaded on this origin that has been identified, it creates two replication forks that move in opposite directions. So that's where the bidirectional replication comes in. Because it's a cycle, the DNA itself is a cycle in prokaryotes. It's cyclic, meaning when we open, one strand is reading 5 prime, 3 prime. As you proceed to the right, meaning the other strand will read 3 prime, 5 prime. But if you proceeded to the left, the strand that was reading 5 prime, 3 prime can actually read 3 prime, 5 prime. And the one that was reading 3 prime, 5 prime can actually become 5 prime, 3 prime on the right. So we have both left and right direction, opposite directions. There is DNA replication. And this is bidirectional replication. So let's move to the next step. We have step 2. Since the initiation complex has been loaded, okay, so this is the supercoiled DNA on this image. Let me just explain on that image. What I was explaining is actually on this diagram here. So we have the supercoiled DNA. This is a double helix. So the topoisomerases can actually cut and join the DNA so that it becomes uncoiled. It shouldn't have those supercoils. And then the origin of replication identified here, we are looking at an area that is rich with A and T. Then the DNA proteins shown in green here using ATP. Remember, they have to be activated. If they're not activated with ATP, they're not able to bind to DNA. And so we have the loader DNA-C bringing it onto the site on the DNA, the uncoiled DNA. So here we have the brown structure trying to show us how these bind at these specific sites so that we can have a complete initiation complex. A typical question could be identifying the components of the initiation complex in prokaryotes. Everything we are seeing here. So we have the uncoiled DNA, with an identified origin of replication. We have the DNA. We have that is activated by ATP. Then some options may even include the DNA B or DNA C. So all those are important. Now, let's move to the next slide, please. So the next, we are looking at step 2, which is denaturation. So when it comes to denaturation, it can also be called what? Unzipping. So the helicase opens the double helix at this replication fork that has been identified at the origin of replication. So denaturation or unzipping simply means the DNA is being opened up. So let's move to the next part. Let's look at the key players. Who are the key players in this denaturation or step 2? So we have the first player, that is DNA helicase. DNA helicase, also known as DNA B. So DNA B is a ring-shaped hexameric enzyme, meaning it has 6 subunits that are arranged in a ring around the DNA strand. Hexa means 6. So it has 6 subunits, and it moves along the lagging strand template in the 3', 5' direction. When it's doing that, it begins to break the hydrogen bonds between the base pairs at a rate of approximately 1,000 base pairs per second. That ticking of a clock, a second arm, it would have moved 1,000 base pairs. So it's that fast. So as it is opening, remember the opening or melting of the DNA for this strand, it's moving in the 3', 5' direction, meaning it's breaking the hydrogen bonds between the complementary what? Bases. And so it's because it's happening at this high rate of approximately 1,000 base pairs per second in E. coli, it means that there's approximately about 1 to 2 ATP molecules per base pair broken. So imagine how much energy is being used just to break the hydrogen bonds, the 2 hydrogen bonds. So we have about 1 to 2 ATP molecules per base pair broken. So this enzyme, helicase, must break hundreds of thousands of hydrogen bonds to replicate the entire E. coli chromosome. So we are the size of the chromosome, how many base pairs in that chromosome. It means each one of those bonds between the complementary bases has to be broken if it is to be what? Synthesized or replicated. So the second player is topoisomerase again. This one prevents positive supercoiling. Now, as we discuss supercoiling, we'll come across positive and negative supercoiling. So here we are looking at the positive supercoiling. Why? Because as the helix unwinds at the fork, because it's unwinding the DNA at the forks, behind the fork, we expect that there's going to be accumulation of supercoils. So we have positive supercoils that accumulate ahead of this fork. So imagine pulling apart 2 intertwined ropes. Actually, this is a very good example. Imagine if you had 2 intertwined ropes and you have to pull them. So you see it becomes tighter. Ladies, as you twist hair and then you pull, you find that it's becoming tighter. So those smaller coils that are being formed is what you should be imagining is happening to the DNA. So apart from intertwined ropes, you actually create more twists at one end. And so if these are left unchecked, it means that the overwinding would physically store the what? The replication fork, because it becomes tighter and on one end you have more coils. So it will hurt, or it will make the whole replication fork to be slow. So DNA gyrase itself continuously relaxes these positive supercoils ahead of the fork. And there's actually a clinical relevance to this activity in DNA. So the clinical relevance is that we have certain antibiotics some of us are familiar with these antibiotics. We have, like, if you've ever heard about ciprofloxacin, fluoroquinolines, kind of. They work by poisoning DNA gyrase and topoisomerase in the bacteria. So this prevents them from reproducing because they cannot synthesize new DNA. So they will not be able to do what? To continue synthesizing. So the next players are the single strand binding proteins, the SSBs. The SSBs or SSB proteins, once separated, the single strands themselves will spontaneously reanneal, meaning they can still reform hydrogen bonds. Because when you just put A and T apart and you let them to snap back, they'll still form the hydrogen bonds. Remember, we discussed hydrogen bond formation. So they can still form hydrogen bonds. So because we want to prevent the reformation of hydrogen bonds, the single strand binding proteins will coat and protect the single strands. So this keeps the DNA strands apart as it unwinds. And because they are apart, it means that it's going to prevent secondary structure formation until each strand works as a template to produce new strands. So the single strand binding proteins are very important in this case. So they are purely structure and they have no enzyme activity. All they do is to just bind on the DNA that is single stranded. It has been unwound, separated, so that it doesn't go back to double strand as a secondary structure. And as they are holding on to the single strands or the separated DNA, they are displaced as DNA polymerase advances as we start the synthesis. When the DNA polymerase come now to produce the new strands, these single strand binding proteins can easily be displaced. So always remember, you can try and think about the SSBs as molecular clamps. So they are molecular clamps holding the unzipped strands open for the polymerase to read that, okay, there is a G here. So on the new strand, I should add a what? I should add a C. So this, okay, so it's good. Taya, you're doing a great job. So he's just showing what I was explaining. So we have the DNA single strand there, and we have the preparing the complex and the helicase and everything. So since this part of the DNA where the helicase is open, the single strand binding proteins have to bind to these opposite strands to make sure that they don't come back together until the DNA polymerase touches. Now, in this picture, you can actually see the arrow showing direction of denaturation. So where the helicase is going, that is where denaturation is happening. But if we can still have helicase on the top strand, meaning denaturation will go in the opposite direction, in the right side direction. So this is actually unidirectional. But for the sake of simplicity, in this image, we're just showing what is happening on one strand in one direction. So we can now move to, we have 4 minutes to go to the third step, which is the priming. So I mentioned yesterday that we shouldn't be thrown aloof when we hear about RNA primase in DNA replication. Because once the template strands are exposed and stabilized, we need a starting point for DNA synthesis. So this is where RNA primase comes in. It's a biochemical problem that only RNA primase can do. Now, most enzymes end with ASE, right? So we know when we say RNA primase, then it's something to do with an enzyme. What does it do? It synthesizes sites, short sequences of RNA that can be used to prime the DNA template so that the DNA polymerase can find somewhere to start from. When we look at replication, it doesn't just start de novo unless it's in a test tube. But even in a test tube, we synthesize primers that we use in PCR and all these other molecular techniques. So for the polymerase to start synthesizing new strands of DNA, they need primers. So that is known as priming. So formation of RNA primers itself, like I mentioned, the fundamental problem is that the DNA polymerases can only add nucleotides to a pre-existing strand which has free 3' hydroxyl group. So that's why even the direction of the growth will be increasing on this 3' end, meaning the new DNA is going to be growing 5' going towards the 3'. So there is no DNA polymerase that can actually initiate synthesis from scratch, de novo, no. So this is an absolute biochemical rule. So no exception is known in any organism unless maybe someone will, in the near future, will actually discover something. But even the ones, like I mentioned, we use in the lab, in the techniques and all, they still need primers. We design primers. Now, the solution is that the RNA primase, the enzyme itself, is specialized RNA polymerase that can initiate a new chain from scratch. But for DNA, we don't have DNA polymerases that can do that. So in nature, a RNA primase is actually used to synthesize a short RNA primer, which is approximately 10 to 13 nucleotides long in E. coli. And so this primer is actually complementary to the DNA template in the 5', 3' direction. As we study this and preparing for the test and the exam, make sure you're always correct on the directions. So when the primers are being synthesized, they are short RNA, approximately 10 to 13 nucleotides, and they're complementary to the DNA template, which is going to be used to synthesize what? New DNA. So these primers are complementary because they have to attach to the template and leave a 3' hydroxyl group hanging so that the DNA polymerase is able to use that 3' hydroxyl group to elongate it. And then later at proofreading, the primer will be removed. And then a complete DNA is going to be synthesized. So now, the short RNA primer provides that 3' hydroxyl group required by the DNA what? Polymerase 3. We are looking at polymerase 1, polymerase 2, and polymerase 3. Make sure we know exactly what each one of these DNA polymerases is responsible for. So for elongation purposes, the DNA polymerase 3 is responsible to make a longer chain. Now, the number of primers is dependent on the template being used. So when we look at the number of primers, remember I mentioned that we have a leading strand and the lagging strand. So when you talk about the leading strand, only one primer at the origin is needed. So once the origin is identified, there's unwinding, then only one primer is added because it's leading, it will be continuous. And the template that can give us a leading strand is a template that is 3', 5', because the primer therefore is going to be 5', 3'. And since the DNA polymerase needs the hydroxyl, so the leading template strand, the 3', 5', is the one used to produce a leading strand, which will be 5', 3'. So only one primer is needed. And then this primer can be used to synthesize a continuous strand of new DNA throughout or until the forks meet, since it's bidirectional, it's in both directions. But when it comes to the lagging strand, the template is 5', 3'. But the DNA polymerase 3 can only synthesize in 5', 3' or so, meaning it needs to read a 3' hydroxyl group end, but then the template is already ending like that. So how can it produce an opposite strand that is going to be 3', 5'? So it does this by producing short sequences. It can revert into short sequences, shorter sequences than the leading strand. So the newly synthesized DNA on a lagging strand is not continuous. It is formed in shorter sequences, which are referred to as what? Okazaki fragments. As a result, it needs more than one primer. So on the lagging strand, you have more primers needed. So there's a new primer needed for every Okazaki fragment. So if the DNA polymerase produces 100 here, 200 base pairs long sequence, 300 base pairs, then 10, and things like that. It means at each point, that gap, it has to be filled by a primer. So at each point, it needs a new primer. So since Okazaki fragments can range in numbers depending on the size of the genome and the sites that can be identified as the start sites, the AT-rich sites on the lagging strand, they would be dependent on how many Okazaki fragments are there. And it's one primer per about 1,000 to 2,000 nucleotides. So this means hundreds to thousands of primers per replication event on the lagging strand. So RNA primers are removed after synthesis by the DNA polymerase 1 using its 5' 3' exonuclease activity. So this enzyme is able to proofread and see that these are RNA. Remember the difference between RNA and DNA. Even in the bases, we have uracil in RNA. And so in some primers, you may actually find that they will contain a uracil. And then even the type of sugar used, you have ribose in RNA in the nucleotides instead of deoxyribose. So the RNA polymerase can identify these differences. So the DNA polymerase, DNA POL1. So it's able to actually remove those primers that were being used. So on the lagging strand, it will remove all those short fragments. Whilst on the leading strand, it's just the very first primer that was added at the origin of replication. Now, I want us to look at step 4. Step 4, we are simply looking at leading strand synthesis and lagging strand synthesis. Leading strand synthesis and lagging strand synthesis. So I'm going to give you an exercise. It looks very stupid, but if you do it, it will help you understand. I don't want to rush these two because they're actually a basis even for protein, for transcription, as well as actual translation that we are going to do. Because you have to identify which one of the DNA strands is going to be used as a template for transcription. And the principle still applies. Is it the lagging strand or leading strand? So we need to identify this. So I don't want to rush this. Now, the homework I have for you is make sure you have a blue, a red, even a different colored thing, so that we identify how the parental strand will be, how the new strands will be, and following the principles of base complementarity, you should be able to identify which one is the leading strand and lagging strand. Then additionally, I'll need you to also identify where you can put each one of the enzymes you've discussed so far from step 1. And then on step 4, where do you think the helicase will be facing? Where is the replication fork? So these exercises, I'm not going to mark them, but they'll actually help you to understand. So I'll be sending them to Chaya, I think in the next 10 minutes. So you if you can print them where you are, it's even better. If you can't, you can just try to work around them in soft copy. If you can copy in your book and try to answer the questions. This will help our understanding, because we are meeting tomorrow at, is it 11 hours, if I'm not mistaken, so that we look at the leading strand synthesis. How is it synthesized? And how is the lagging strand also synthesized? We have key points that we need to really understand. Okay, unless we have some questions or something. Okay, so silence means all is well. Thank you very much. So let's meet tomorrow. I think it's 11 hours, right? So let's meet tomorrow 11. Okay.